today's video is about biological molecules. So basically today we are going to discuss those biological molecules that are the part of our nutrition that breaks down in human body, and these nutrients are considered as the essential nutrients that are required for a human body to live. So we'll start with carbohydrates, proteins and fats, and we'll compare them on the basis of these characteristics. Number one is chemical elements. Number two, we are going to talk about the types. Number three, we are going to talk about their basic unit, the structural unit of them, or we can also call it as monomer. Then we'll discuss their functions, how much energy they give, what are the diseases, and what are the food tests that can prove them. So chemical elements of carbohydrate, proteins and fats are same as carbon, hydrogen and oxygen. So the difference comes in the presence of nitrogen, which is only present in protein, whereas all of the fats, carbohydrates and proteins, they have carbon, hydrogen and oxygen. Okay. Now we'll talk about the types. Carbohydrates exist as monosaccharides. The example of monosaccharide is glucose. Then we have fructose, galactose. The disaccharide has examples as maltose, lactose, and sucrose, whereas polysaccharide starch, glycogen, and cellulose. If I'll simplify them, we can say that fructose is present in sugars and they can give you energy, whereas polysaccharides are basic for storage. You do know that cellulose is responsible for making the cell wall, and the disaccharide lactose is there in the milk. If we talk about proteins, proteins have the essential amino acids which are responsible for their formation. They exist in the form of globular proteins and open chains polypeptides. Whereas fats, they exist in two forms, saturated and unsaturated. Saturated fats are mostly solid at room temperature, whereas unsaturated fats are liquid at room temperature. Now we will look at the structure of glucose. Glucose is having a very hexagonal structure in which you can see 1, 2, 3, 4, 5, 6 glucose units, and the formula is C6H12O6. If we talk about the structure of an amino acid contains a carboxylic group and an amino group, whereas fats has this basic structure in which there are 3 fatty acids and 1 glycerol. Okay, so glycerol can be broken down, and then they are the substances that are formed when they are fully digested form. When carbohydrate, proteins and fats are fully digested, these are the end products that are formed after digestion. Okay, so now we'll talk about their functions. First function of carbohydrate is that they are responsible for respiration. Respiration is the basic function of carbohydrate. Now we do know that carbohydrates are they are used for storage. They are also responsible for making cell walls, so they have their function in this regard as well. If we talk about protein, protein is responsible for growth, repair. They're also responsible for many vital functions, and then there are transport proteins that are also present in cell membrane. If we talk about the structure of fats, fats have oily layer. They form the oily layer outside the plants. They prevents evaporation. They are also responsible for insulation. They are also responsible for forming the adipose tissues. If we talk about the energy, fats give maximum energy, that is 39 kilojoules per gram, whereas carbohydrate and proteins gives less energy per gram. But the carbohydrates, they give efficient energy. They are soluble in water, whereas fats are insoluble in water, and if we want to take energy from the fats, it takes time. You can see some examples of all these carbohydrate, proteins and fats. You also know that we are been consuming these things, right? If we want to test that whether the fat is present, whether the carbohydrate is present or not, we do Benedict test. In Benedict test, basically the red color is formed when there is a strong amount of glucose present. The blue color of Benedict solution changes into red color, brick red color. It also forms yellow-orange color in between. Then we'll talk about the Burette test. In Burette test, you detect the presence of protein, which converts blue color Burette solution into violet color. Whereas in fats, we take the oily thing or the butter or anything in which you want to test. We put it into alcohol, we dissolve it, and then we form a fumation layer on the top of the water, which shows that whether the fats are present or not. There are some diseases like diabetes, kwashiorkor and obesity, which are particularly related to each of the particular nutrient. You can find about them in detail as well. So this was all about carbohydrate, protein, and fats. Thank you so much.